So hello students, welcome back to your own channel VA NEET Academy. So today's topic will be nucleic acid. So we have till now covered the biomolecules like carbohydrates, proteins, lipids, and today's will be nucleic acid, and this is followed by enzymes. I think the final biomolecules of our syllabus. And being a biotechnologist, this is one of my most favorite topic on nucleic acid. And all of you know the role of a nucleic acid, which is DNA and RNA, isn't it? So DNA carries the genetic information, RNA helps to express this genetic information into traits, converting the genetic information, the code present in the DNA, to transcribe and translate into the expressive proteins so that it can be expressed what it carries. The information is translated with the help of a RNA. So beautiful topic, and hope you are clear with the topic today after going through the video. And any doubts, do comment and ask me. Do subscribe the channel and share with your friends so that this will help me motivate if I get more and more subscribers. Thank you students, and keep watching, keep the bell icon ready so that you get the uploaded video immediately as I do it. Thank you. So students, today we will start our class with nucleic acid. Two types you have, DNA and RNA, all of you know, deoxyribonucleic acid and ribonucleic acid. Okay, so let us start with our definition. Definition of a nucleic acid. So how will you define a nucleic acid? Okay, so I am writing nucleic acid in short as Na. Okay, nucleic acid. So nucleic acids are the polymers. Okay, so they are the polymers of nucleotide. Okay, they are the polymer of nucleotide. So many nucleotide, they join together, okay, to form the nucleic acids. Okay, nucleic acids are the polymer of nucleotide. Okay, and what is their role? Their role is they play a big role in carrying the genetic information, transferring the genetic information from one generation to another, and also expressing the genetic information. Okay, so this is about the definition of a nucleic acid. Let us now come to the components of nucleic acids. Nucleotides. Okay, you know what is a nucleotide now, isn't it? Nucleotide is a single unit of a nucleic acid. So when many nucleotide combine together, you get a nucleic acid, isn't it? Clear, right? So what are the components of nucleotide? Nucleotide is made up of nitrogenous base. Okay, nitrogenous base. So hereafter I will write in short nitrogenous base as N2 base. Okay, so wherever the video has N2 base, it means nitrogenous base. Okay, it has pentose sugar. It has pentose sugar and it has a phosphate group. Okay, so these are the three main components of a nucleotide. Nucleotide has a nitrogenous base, a pentose sugar and a phosphate group. Okay, so basically a nucleotide, a schematic diagram if you write, you will write it as it is made up of sugar, nitrogenous base and then phosphate attached to it. So this is a one single nucleotide. Okay, this is one single nucleotide. There is one more term called as nucleoside. Nucleoside. What is a nucleoside? Nucleoside is just a sugar and a nitrogenous base. Nucleoside is made up of only sugar and nitrogenous base. So what is absent? The phosphate group is absent. So when a nucleoside is attached to a phosphate group, you get nucleotide. Okay. So, do not get confused students, nucleoside SSIDE means sugar and nitrogenous base. Just remember, S is there, nitrogenous base is there, sugar is there, nitrogenous base is there. Tide means you have an addition of phosphate, nucleotide. Okay. So, nucleoside when attached to a phosphate is equal to, what is it? Is equal to nucleotide. Clear? Okay. Hope it's clear students. Okay. So, this is about a nucleotide. Now, we will see in detail the sugars. I told you the sugar present is a pentose sugar. Pentose means what? How many carbon atoms? Pentose is a 5 carbon sugar. Okay. So, the sugar pentose, you have two types of nucleic acid, isn't it? The DNA and the RNA. Okay. So, DNA is deoxyribose nucleic acid. Okay. RNA is ribose nucleic acid. So, what is the difference between DNA and RNA? There is a difference in the sugar. In DNA you have a deoxyribose sugar, in RNA you have a ribose sugar. Clear? Okay. So, what is a pentose? I told you it is a 5 carbon component. Okay. So, if I draw the 5 carbon component, you will be drawing the structure something like this. Okay. So, C1 prime, C2 prime, C3 prime, C4 prime and then C5 prime. This is the 5th carbon. So, 1, 2, 3, 4 and 5 carbon ribose sugar, pentose sugar. Okay. So, this is number 4. I will write it here, number 4. Okay. So, here this is H, this is OH, this is H, this is H. Okay. So, this is your deoxyribose sugar. This is your deoxyribose sugar. Okay. Now, how will you draw a nucleotide in this? Now, in this, if you want to draw a nucleotide, you need to see that the C1 prime, see I am writing the numbering as 1. If you add a small dash above, you call it as prime or else 1 dash. Some people call it as dash, but I prefer saying it as 1 prime, 2 prime, 3 prime, okay, 4 prime, and 5 prime. Okay, this is 1 prime sugar, this is 2 prime sugar, this is 3 prime sugar, this is 4 prime sugar, this is 5 prime sugar. Okay, carbon, right? Okay, so there is a 5 carbon atom in a deoxyribose sugar. Okay, now we need to attach a nitrogenous base to this structure. Nitrogenous base is always attached to the 1 prime carbon of the sugar. So, here you can add a nitrogenous base. So, if you add a nitrogenous base to the 1 prime carbon, you have an addition of nitrogenous base to the ribose sugar. Okay, and then what else has to be added for a nucleotide to be formed? A phosphate group. Now, can you say this is a nucleoside? Yes, isn't it? The sugar, the nitrogenous base. Now, I need to convert this nucleoside into nucleotide. So, I need to add one phosphate group. Now, where is this phosphate group added? It is added to the 5 prime carbon of the sugar. So, here I will get a 5 prime carbon is attached to the phosphate group. Now, this is the phosphate group, isn't it? This is the phosphate group. So, now the sugar is there. Okay. So, now this is the sugar, deoxyribose sugar, the nitrogenous base and the phosphate group. Now, this is your nucleotide formed, isn't it? Okay. Now, we will see the difference, the slight difference in a ribose sugar. Okay. So, here the ribose sugar, again it is a 5 carbon sugar structure. So the same ribose sugar, okay, C1 prime, C2 prime, C3 prime, C4 prime, and this is C5 prime. Okay, all the 5 carbons. Okay, so this is H, this is OH, this is OH, this is H, and here the nitrogenous base, N2 base, and here the phosphate group. Clear? So what is the change? Have you noticed? You have noticed the change, isn't it, between the sugar in the DNA and the RNA? Okay, so can you see the 2 prime carbon has only hydrogen here, and the 2 prime carbon has a hydroxyl group in a ribose sugar. So that is the difference. And now you will remember what is the name of this. This is a deoxyribose sugar. So what has happened? The oxygen at the 2 prime end has been removed in the DNA, and this is why it is called deoxy. Okay, deoxyribose. Ribose is like this, where the 2 prime hydroxyl group is present. So when you remove this oxygen, you call it as deoxy, isn't it? And when you remove this oxygen, you get a DNA called as deoxyribose. Hope it's clear, students. Okay, very simple. Just understand the logic. Okay, don't mug up things, just understand the logic and you will understand everything clearly. Okay, so this is about a nucleotide and the structure of a sugar in a nucleotide. Okay, now we'll come to the nitrogenous base. The nitrogenous base, I am not going into a very deep chemistry of a nitrogenous base, because I don't think for 11th class students you need to understand all that. Okay, so I will just introduce you what is a nitrogenous base types. Okay, so nitrogenous base are basically 2 types. Okay, purines and pyrimidines. Purines and pyrimidines. Okay, so purine is a double ring structure. That's it. I am just going into this limit. Okay, so purine is a double ring structure, pyrimidine is a single ring structure. Okay, so when I say this, roughly the skeletal diagram, I can say that it is something like this. This is purine having 1 and 2 ring. Okay, and this is pyrimidine having just 1 ring. Okay, this is the skeletal diagram of a chemical diagram of a purine and pyrimidines. Okay, so now purine in turn, purine has 2 types. Okay, adenosine, shortly written as A, and guanosine, shortly written as G. Okay, right? And pyrimidine is of 3 types. Okay, cytosine, shortly written as C, thymine, shortly written as T, and uracil, shortly written as U. Uracil U. Okay, so purine, adenosine, guanosine, A and G, pyrimidine, cytosine, thymine, and uracil. Pyrimidine is a single ring structure, purine is a double ring structure. That is, I think, more than enough for you right now. Okay, for this to understand. Okay, so now we will understand the base pairing rule of a nitrogenous base. Base pairing rule for a nitrogenous base. Okay, always understand that purine always pairs with pyrimidine. This is P is short form of purine, PY is short form of pyrimidine. P has to pair with only pyrimidine. Purine has to only pair with pyrimidine. So in that case, if adenosine is the purine, okay, it has to pair with T, that is thymine, okay, and if guanosine is the purine, it has to pair with cytosine. Clear? Okay. A pairs with T, G pairs with C. And how is this pairing happening? This pairing happens with the help of a hydrogen bond. This pairing happens with the help of a hydrogen bond. Adenosine A pairs with T with the help of double hydrogen bond. G pairs with C with the help of a triple hydrogen bond. Okay. So, the pairing happens with the help of hydrogen bond. Okay. This pairs with a triple hydrogen bond, H bond, and this pairs with a double hydrogen bond. A pairs with T and G pairs with C. Okay. A T has a double bond, G C has a triple bond. Okay. Now, there is a slight difference between a DNA and RNA. Okay. The difference is that DNA contains only thymine and cytosine as a pyrimidine. Okay. RNA has only uracil and cytosine as a pyrimidine. Okay. So, basically DNA has thymine and RNA has uracil. RNA does not have thymine. So, in that case, in RNA, if adenosine is the purine, it pairs with uracil and not with thymine. That is the difference. If it is an RNA, A pairs with U and not T. Got it? That is the only difference. Okay, right? So, this is the pairing rule. Okay. So, now we will go into the final topic, which is your structure of a DNA. Structure of a DNA. First, we will understand the structure of a DNA. Okay. Now, the structure of a DNA was first proposed by Watson and Crick, a very famous scientist, Watson and Crick. Okay. Watson and Crick in the year 1953. In the year 1953. So, they proposed a double helix structure. They propose a double helix structure. So, that means according to them, DNA is a double stranded structure. Okay, and these are double stranded structure are joined together with the help of a nitrogenous base. So, if it is A here, it has to be T. If it is G here, it has to be C, G, C, T, and then A, something like this. Okay, like a ladder kind of a thing. Now, when he says it is a double helix, now this ladder twists like a double helix, something like this. Now, this becomes your DNA. Okay, and they run in an anti-parallel direction. So, that means if one strand ends at a 5' carbon, the other strand, so one has a 3' end and the other has a 5' end. So, suppose this is the one strand, okay, with 3' end and 5' end, the other will be in an anti-parallel direction. That means this will be 3' end and this will be 5' end. That is what it means by an anti-parallel strands. So, if this is 5', this is 3', and if this is 3', this is 3' and this becomes 5'. Okay, now you know what is a prime. Okay, so these are the number of carbon in the ribose sugar. Okay, so this is 5' end which is attached to, remember where? The phosphate group, and 3' end attaches to the OH group. Okay, just go back to the diagram and check 3' will be attached to an OH group. Okay, so this is a free 3' carbon and this is a free 5' carbon. That means this does not have a phosphate group and this has a OH group. Don't get confused, students, not required for you for the 11th class student. Okay, so that is about the structure of a DNA. Clear? Okay, so now about the structure of a RNA. So what is the structure of a RNA? Very simple. Okay, structure of a RNA is not complicated as this, because it is a single stranded structure. It has only a single strand, so it is just like this. DNA is double stranded structure and hence it has a double helical formation, whereas RNA is a single stranded structure. Okay. Now basically you have three types of RNA, you know, right? The mRNA, which is called as a messenger RNA, okay, mRNA, then tRNA, which is a transfer RNA, okay, and then the third type is mRNA, tRNA, rRNA, which is a part of a ribosome structure, okay, which is present in the formation of a ribosome. Sorry, ribosome, not ribose. Ribose is a sugar, okay, and it is involved in the formation of a ribosome organelle, you might have seen, okay. So this is about the structure of a RNA. Clear, students? So now we will see what are the functions and then conclude the class with a slight comparison between the two features, okay. So what is the function of the nucleic acid? Function of nucleic acid, all of you know, it has a role in it carries the genetic information, okay, it carries the genetic information, right. So it also transfers the genetic information, right. It only not carries it, it also give it to the next generation also. And third role is it controls all the cellular activities. So everything is a mastermind. I mean, all the mastermind of our cell is a DNA and RNA, that is nucleic acids, okay. So these are about the function of a nucleic acid. So now let us see the final portion of the video, comparison table. So you have a feature, okay, feature present in DNA and the same feature present in RNA. Let us try and see, and let us see if you can answer these. So feature sugar, first feature sugar. What is the sugar present in DNA? Deoxy or ribose or only ribose? It is deoxyribose. Now you know why it is called deoxyribose, isn't it? And then the sugar present in RNA is ribose, right? Correct? And what is the base present in the single base change in DNA and RNA? In DNA it is thymine, rest all is same, right? And in RNA it is uracil, rest all is same, okay. So there is no thymine here, there is no uracil here, okay. And next we will see the strands. How many strands are there in DNA? Double, and here it is single, okay. Now the function, what is the function of DNA? It carries the genetic information, right. And what is the function of our RNA? You might have seen it has a role to play in protein synthesis. It has a role to play in protein synthesis, okay. Hope the class was clear, students. I can talk more and more about nucleic acid. This is one of my favorite topic, but then it's 11th standard class. Do let me know if you want to know more about it, a detailed structure and etc. Okay. So thank you for watching. Keep watching our channel. Do subscribe, like and share, and do comment your suggestions and doubts as well. Thank you, students.